I just wanted to explain the outline for the evening and what we'll be talking about. So first, I'll quickly explain my research background.
Oh, check your mic.
Is that better? Perfect. Okay. So first I'll explain my research background, and then we'll go into a little bit of background and information that you need to know to understand what we're working on. So what are microbes, what are antibiotics, and what is antibiotic resistance? Then we're going to be talking specifically about the research I've been doing around here at the beaches and around WHOI, and then the larger significance of that. So to sort of frame what I've done, the past research I've done as well as my current research, I think of it in this framework. So I'm interested, and I consider myself an applied environmental microbiologist. And I'm interested in these three categories in projects that envelope these three. So working in aquatic environments, which right now is the ocean on beaches, looking at microbes that live there, and then working on questions that relate to public health. So I got into this. When I was younger, I did a science fair project on bacteria, and I really, really enjoyed it. And so then as I was looking at where I wanted to go to college, I decided I wanted to pursue a degree in biology at DePauw University. So this is a small school, liberal arts school in Indiana, not anywhere close to the ocean. However, this going to DePauw allowed me to get started doing research early on, and I found out that that's what I really enjoyed. And it gave me a lot of opportunities to pursue research related to bacteria. So on the top picture here, I'm sampling with one of my former advisors in a pond in Indiana, and we were looking at the bacteria there in those different ponds and how they changed over time. Here I'm studying microbial decomposition of ecological dry toilets in Ecuador. And then this picture here was after a summer in Maine doing eelgrass conservation as well as cruise ship monitoring. So through my experiences at DePauw, I was able to get a lot of different research experiences and figured out exactly what I wanted to do. So as I was applying to graduate school and deciding what I wanted to do for the next 5 years of my life, I still thought back to that same framework, and I wanted to work on projects related to that. So I decided to come to the MIT-WHOI Joint Program in Oceanography and work in Becky Gas Lab at WHOI. So that's how I got to be doing this work, and I'm really excited to have spent the past 2 years working on environmental antibiotic resistance, and to continue spending the next hopefully three, but potentially 4 years doing that.
We'll get down to 3.
Okay, good. Thumbs up. 2 thumbs up for that. Okay, so now that you know a little bit about me, we're going to talk about background information related to the project. So, what are microbes? Has anybody heard the word microbes before? Okay, so pretty familiar with it, but this is the definition that we're going to use for today. So, organisms that can be seen with a microscope. And these are just pictures that I pulled of different types of microbes. These are under microscope magnification, so they're obviously not this big if we're actually able to look at them, or this color for that matter. So these are just 3 different ones and different groups. So the one on the left is bacteria. This one's a type of fungus, and this is an archaea, which we're going to talk about here. So life in general is divided into 3 groups, and biologists think of life in these 3 groups. So we have, can you guys see that? Okay. So we have this group up here, which is bacteria, this group, which is archaea, and this group, which is eukarya. And the main differences between these 3 groups are genetic, biochemical differences, and structural differences. But to point out for the rest of the talk too, bacteria, that group is what I study, and that's what we'll be talking about for the rest of the time. But it's important to know these 3 in general. And then eukarya are cells that are like our human cells. But the archaea and bacteria are different than our human cells. So I have a question for all of you. How much of this tree and how much of these divisions do you think it contains microbial life? Or rather, what ones do you think, what percentage or grouping do you think don't contain microbial life? Any guesses? So you think they all have microbial life in them?
Yeah.
Oh, you guys are great. But I thought we were going to go with a smaller amount. But still, so there's actually two that don't have microbial life. Normally people think there's a lot smaller amount, but you guys are on it. So life is mainly microbes. But the animals and the plants are the two groups that don't have any microbial components. The rest do. So I think that's really cool when I think about microbiology, because that means everything around us are these tiny things that we can't even really see, right? And only recently have we had the technology to look into those and to study them in more detail. So, now that we know that they're all around us, why do we care about them? So there's a lot of different reasons why we care about them and why we study them. One is nutrient cycling and degradation. So they're able to cycle cycle important elements like carbon, nitrogen, phosphorus, and iron, and allow those to decompose and be reused. Also, if you've ever had yogurt, beer, wine, any of those things which are very enjoyable, you can thank a microbe for that, because that's done through the process of fermentation. Humans use microbes for bioremediation, which I'm showing in this picture. So microbes can eat very different things compared to what we can eat, and we're able to exploit those capabilities to engineer bacteria that can eat oil up and clean up our messes. And then we also use it for bioprospecting. So this is sort of going out into the environment and looking at microbes and what chemicals they're producing, and finding out what those do, and then using them for our own uses. So an example of this that we're going to talk about in more detail is penicillin. So that's a common antibiotic that we were able to find and then use for our own purposes. And then
Can you tell us what the size of a microbe is?
What's the
Or how
best
much
way?
power do you need to see it?
So it depends the different size of it, because they can be variant in size, but they're, let's say, like 20 microns, but I can't remember. There's a nice analogy for the size, the amount you can fit into a period at the end of a sentence, but now I can't remember the number. You know what I mean? It's like,
I just wondered whether I could see the microbe or not.
you probably
How
could.
much power do you need?
I'd say like 50x, but it depends on the microscope, I think. It depends what kind of bacteria what kind of microbes you're trying to see, because some can be really large. So hopefully that's somewhat helpful. So we're also interested in microbes because they live in very diverse environments. And they live in all of these environments, and basically everywhere we've looked, we've been able to find microbes. So they're into these diverse environments and have unique ways of living in these. And these are areas that humans would not be able to live in. And to drive that point home, I just wanted to have a little bit of the numbers of microbes in different environments. So as you can see here, in ocean water, we have a 500,000 cells in a mil or a cubic centimeter, which is a really small volume. In ocean sediments, we have even more. So we have up to 220 million cells in that same amount. So that's huge. Then in rivers, we have double the amount we have in ocean water. And then in forest sediments, so those are terrestrial, much less than we have compared to the ocean sediments, but still a lot of microbes around us. And not to gross you out, microbes are all over us as well. They're not just in our environments and on tables and chairs, they're in us. And that's not something to be disturbed or scared about, because they're actually helping us. And your body provides many different environments for microbes. One of the things I like to think about with this is that the bacteria and the microbes on your left hand are different than what's on your right hand, which is weird because you'd think they'd have a similar environment. So people have been studying that and studying bacteria all over your body, and they found different things in every place. Originally, it was thought that humans have 10 microbial cells for every one human cell, which is crazy, right? That means you're mostly microbes as well. Our environments are mainly microbes, and you're mostly microbes. However, as more research has gone on, that number's dwindled to like 3 microbes to 1 human cell, so it's not quite as dramatic, but still a lot, and you're still mainly microbes. And the microbes that are on your body have been implicated in a lot lot of different diseases, including irritable bowel syndrome, diabetes, obesity, ulcers, and they're also the form of cavities, so the sort of the plaque that you get on your teeth, that's from microbes. So I just told you things that they can cause problems with, but in general, they're helping you. For example, they're colonizing your skin, so other pathogenic bacteria can't make you sick, and they're also in your gut and help you digest food. So there's something that you need. And overall, depending on your weight, the weight of a person, your microbes in your body make up about 4 pounds. So it's a substantial amount. And you're shedding them all the time, so you're actually going through them and getting new ones. And this, I just think, is really interesting, sort of to drive home the point that bacteria and archaea are really important. So this is, the top number is the number of estimated bacteria and archaea, so those two groups that are all microbes, that in the entire world. And the number at the bottom is the number of people in the whole world, which I tend to also think of as a huge number. But in the context of how many bacteria and archaea out there, it's barely anything. So that's just important to keep in mind. And in general, they're all helping us out. So the other thing that I think is a misconception about microbes in general is that they're all bad and they're all causing diseases. But that's really, they're the minority of that, so they're not the majority. So here I actually have some pathogenic or disease-causing microbes, and you might have heard of some of these. The one on the left causes tuberculosis. We also have two causing pneumonia and one causing dysentery. So those are big problems, and they are important to learn about, but we can't think of all of them as scary and bad, because most of the time they're good or just neutral. Does anyone have any questions on microbes before we talk about antibiotics?
What are the difference between the bacteria and the archaea?
So they have structural differences as well as genetic differences. And that's sort of been discovered, I'd say, about 40 years ago. So that sort of revolutionized the way we thought about things.
So when you first asked the question, what doesn't have microbes?
Mm-hmm.
And a lot of us thought that they all do. So what doesn't have, because if we have all these microbes, are there some plants and animals that don't have?
Oh, that's a good point. I meant so everything, plants and animals, did everyone hear the question first? So, yes, most plants and animals that I know of that have been studied have microbes in them. So I was just talking about within those groups, we wouldn't consider ourselves as microbes. So plants and so they're just like, they aren't considered microbial, but they have microbes in them. Does that make sense? It's kind of confusing.
Well, we tell different types of microbes in the microscope. They're all small and look alike, and there are so many of them.
So there's actually a lot of different ways that people do it. You can stain them and tell differences, but a lot of the stuff we do now is sequencing, so you can figure out by using differences within those bacteria to figure out exactly what species and type it is. So people do use microscopy a lot still, but there's more advanced techniques in some ways, or advanced ways that you can look under the microscope to tell different ones apart.
You say sequencing, like DNA?
Yep.
Oh, okay.
Exactly. Yep.
Can you give specific examples of the few things that do not have microbes?
The few things that don't have microbes. So I don't study interactions between plants and animals and microbes, so I may not be the best person to ask this question to. I don't think I know of any that people have studied that I've read about that have any. Do you have any thoughts on that, Becky?
So that pretty much everything has some microbes then?
That I know of and that has been studied, to my knowledge. So that's probably not the best answer. But I think in general, everywhere we look for microbes, we find them.
Unless we're looking on the moon, and that would be exciting.
Yeah, that would be very exciting.
Okay.
Yep. And people are trying to do that too. Well, or like Mars, right?
Yeah.
Yep.
They're getting close on, they're getting close
Right.
on.
Yeah, there's been some things where
They
they
found
found water.
evidence.
Yeah.
What about bleakers? A couple containing fluids, do they have microbes?
Leakers? I'm not sure what that is.
Like whiskey, for example?
Oh, I haven't. Yeah, they would, wouldn't they?
They did originally.
Yeah, I guess.
Yeah.
Okay, so now we're going to talk a little bit about antibiotics. And to start off with, I want to talk about a quick discovery of antibiotics. So this is Alexander Fleming, and here he's seen on his bench with some petri dishes. And here I'll show you his discovery of penicillin. So, So here we go. So to start out with and to frame this circle that you see here, if I can find the mouse. Here we go. This circle here is called a petri dish, and it contains food that the microbes like and then grow on. And each one of these little dots is a colony, and each colony contains many, many, many cells, so that you're able to see this with your naked eye. It allows you to study bacteria better. So what Fleming did is he went on vacation. He left his plates out, which is not the best way to go. Then he came back and he saw this weird blob here, this white blob at the top. And he was sort of wondering what's going on. These are the cells down here are what he was trying to grow and what looks normal. But if you can look as we go up on that plate, they look a little bit odd. They're not as white, look like maybe they're not doing so well. And as we get closer up to this blob, there's not really anything there at all. So he thought that there's something going on in this white blob here, some sort of maybe chemical that's going out that's killing off these cells. And he was right. So he studied this for a number of years. He first discovered this in 1928, but it took a number of years to get everyone together and to figure it out. But this colony, this blob, is actually producing penicillin, which was one of the first antibiotics that people were able to use. So now that we know how antibiotics can get discovered, I wanted to just point out a quick definition of what exactly they are. So they're compounds produced by a microorganism, or just a microbe, because those are comparable words, that inhibits the growth of another microorganism. And it's a natural process, but we can also chemically synthesize them as well. And our discovery of these antibiotics has been relatively recent. So on this timeline, they're showing some sort of like pre-antibiotic uses that were available. But as we're going onwards, you can see here's penicillin, as well as a lot of other antibiotics that you may be familiar with, and going onwards into 2003. Now, these are obviously not all the antibiotics that they discovered. They've just chosen a few for this figure. But the important thing that is true is you can see at the beginning, we were getting a lot of antibiotics. And as we're moving into more common times, we're not getting quite as many. And that's part of the problem that they're encountering. It's hard to come up with new targets for antibiotics. And so that's a problem that we've been running into. Though, excitingly enough, they were able to find a new antibiotic in January that was published, and that a lot of work was being done on that one. So we also use antibiotics very frequently. And this was surprising to me when I started studying this, but we don't use antibiotics just for human infections, though that is one way that we use them. We use them more often for animal agriculture, aquaculture, as well as plant agriculture. And in the US, actually 70% of our antibiotics are used for animal agriculture, so it's a really huge amount. But we're not using that all just for sick animals, right? It turns out that if you feed animals antibiotics at lower levels than what you and I would need for an infection, they actually grow faster. And so if you can imagine, if they're growing faster, you can take them to market sooner and potentially make more money. So a lot of farms put antibiotics into the feed to allow them to grow faster. And also it helps for infections too, because if you think about how packed farms can be, like this picture of all these chickens here, you can imagine that if one chicken got sick with some sort of infection, it would spread very quickly throughout the rest of the farm as well. So that's important to keep in mind because it's not, I think in general we tend to think about it as just being for human health, but that's not really where the majority of our use is coming from.
Does that mean we're getting a lot of when we eat for food?
Potentially, yeah.
Yeah.
So there's been a lot more work looking at that because people are concerned about it.
And you said it was a natural process. Would the body naturally heal itself? Form antibodies to counteract the, versus antibiotics and.
Well, the antibiotics only affect the bacteria just because of the way the structure works. So they kill off just bacterial things. So they're not killing off anything in your body. So they don't affect your human cells. They just affect whatever is causing the infection.
But the stuff in your gut, though, and stuff like that has to be replenished.
Yep.
Some of the things.
Yep, that's exactly right. So that's part of the problem with them is if you take antibiotics, right, most of them are not very specific. So they are killing off a lot of your gut microbiome. And that's, some doctors recommend after you take antibiotics, you would take maybe a probiotic, like yogurt, so that you're recolonizing your gut with like good bacteria. Because otherwise the bad bacteria can sort of get a foothold more easier if there's not anything else to take the place. And so that leads well into how antibiotics work. So in general, they target the bacterial cells to stop them from growing or to kill them. And this can occur by attacking the outside of the cell through the cell membrane or the cell wall, or by basically stopping or hurting the processes within the cell. And there's lots of different ways that antibiotics can do that. But we're not going to get into all of them today. So does anyone have any other questions about antibiotics before we move into antibiotic resistance?
What created the antibiotic? I mean, what made it happen?
So that's a big question, actually. So they've been around since way before humans got involved. But one of the reasons that it's thought about is it could be that the bacteria are secreting these as chemical signals. So maybe they're sending signals to bacteria that are like them, like, hey, I'm here. Or maybe they're sending a signal to other bacteria sort of to get away or to kill them. So it's also thought as like a biological warfare. So like, okay, maybe this one bacteria is secreting these antibiotics to kill off other bacteria around them so that they can have that environment for themselves. But that is still being studied, and it might be a variety of those things.
Question about this, example, about chickens which got sick, but we have the disease which, and bacteria which have nothing to do with humans. You cannot get sick with the same disease, for example. So antibiotics, they only affect those bacteria and probably do not have any effect on humans.
Well, the problem partially, especially in the US, in the EU, they actually have more legislation about this. But to this point, you can use the same antibiotics that you would use in humans in animals here, so that's kind of problematic and allow for resistance overlap. But a lot of our antibiotics aren't very specific. So even if it's some bacterial infection that's in chickens, that wouldn't be in humans, the resistance that would emerge can pass to humans too.
You mentioned the probiotics versus the antibiotics. I understand that in other parts of the world, when you have like chronic bowel diseases like ulcerative colitis, people actually do fecal transplants to get the right kind of bacteria growing in your gut, and that's an alternate way to treat from what we do here in the U.S. So we give people all these sulfa drugs.
Yeah, that's a good point. So he's talking about fecal transplants, and that's one of the new emerging technologies. And sort of the idea is that if you have the wrong microbiome that's causing some sort of issues with the microbes that you have within you, that then if you put the right ones in there, your symptoms go away. And that actually works really well for people suffering from various illnesses. Any other questions?
That doesn't mean you should go out and recruit.
Yeah. Good clarification there.
That's what the dogs do.
Okay, so now we're going to talk about antibiotic resistance. So resistance is when a bacteria is not affected by a particular antibiotic. And this is like antibiotics themselves are a naturally occurring process. And some research has gone into this, which I think is really interesting. So they've gone out and looked in environments that haven't really had contact with humans, like ancient sediments, very isolated caves, and most recently an isolated tribe of humans in South America. And they found antibiotic resistance there. So that sort of drives home this point that it's natural and it's not something that's completely human caused, though it's likely strengthened by the amount that we're using because we're using so much. So now you might be asking, how can bacteria resist antibiotics? So I made this little schematic that gets at that. So if you'll imagine with me that this is a bacterial cell, and these little orange dots are antibiotics. And so there's a variety of things that could happen, and I've just picked 4 to show here. But the antibiotic could get into the cell, and then it might not do anything. So that's showing immunity. It's also possible that if the antibiotic, let's say, affects the black triangle, that the cell could change the target from a black triangle to a blue triangle, and then the antibiotic wouldn't be able to affect it, so that allows them to resist the antibiotic. Another way is to just take the antibiotic when it's in the cell and pump it back out, so it's outside of you and can't hurt you. And then finally, if it gets in, you can just inactivate the activity. And these are just, again, just a few of the many ways that antibiotic resistance can occur.
How do you inactivate the activity?
There's a bunch of different ways they can get it in. It depends on the specific antibiotic and what it's doing in the cell. Okay, so now that we know that resistance is out there, how can it get spread? So this is a diagram from the CDC, and it's showing of selection of resistant bacteria by exposure to antibiotics. So here we're starting out with blue bacteria that are sensitive to antibiotics, and the purple magenta color that are resistant, right there. So then if we treat ourselves with, let's say, a human with antibiotics, then what's going to happen is the ones that are sensitive are going to die off, so that's what these little X's are showing. And then the resistant ones are going to still be alive, right? So they're going to stay around. And now, because all the other ones are gone, there's not as much competition, they're going to be able to take over and there's going to be a lot more of them. So that's selection, what selection happens by exposure to antibiotics. But this last one is showing that the bacteria can share resistance. So they can, one resistant bacteria can take it and share it and pass it to another one. And that can happen between different types of bacteria. So that's problematic in many ways. And then we'll talk about that in a little bit more detail related to the beaches. So now that we know about antibiotics and antibiotic resistance, I want to pull this all together into the marine environment. So starting off, we think of the ocean as a reservoir for resistance. And we think of it because it has 2 separate inputs. One is resistance naturally occurs in the environment, as we talked about, so it's already there in normal marine bacteria. But we also have human inputs that bring in antibiotics through various wastes that we have. So remember when I showed you that picture of aquaculture, agriculture, human inputs, and plant agriculture? So all of those processes can produce waste that then can get into our environments. And then potentially, even if it's just going into wastewater treatment or getting into streams, and then rivers, and then potentially into the ocean. And with aquaculture, the antibiotics are already directly going into the ocean as it is. So that's what's happening there. And the resistance, as well as pathogenic bacteria, can be coming in through that human-caused inputs. And so with those 2 inputs together, what happens is it allows for resistance to be shared in the environment. So the regular marine bacteria may or may not have resistance, and these new bacteria coming in, or resistance coming in, can then swap, and they can share in the environment, because you're putting these two things together that wouldn't normally necessarily all be interacting. Does that make sense? So it's kind of, I know it can be kind of confusing. And so this causes the public health concern, because it could be that the pathogenic bacteria, when they're in the beach or oceanic environment, pick up a resistance, or they could, pathogenic bacteria could be sharing it with their resistance with marine bacteria that are already there. So, if they're there, does that matter? Are humans impacted by that? So there's actually two ways that we think antibiotic resistance can be transferred from the ocean. So one is through consumption of various marine goods, like shellfish, and others through recreation, fishing, boating, etc., swimming, where sometimes you're either interacting directly with the water through your skin, or usually when you're doing these things, sometimes you ingest water as well. And so these two ways allow the potential transfer of resistance from the marine environment to humans. And then going back, when we interact with the ocean environment or through our waste products by aquaculture, agriculture, human waste, etc., those can get back into the ocean as well. And also it gets in directly because we go, as you bathe in the water, you're sort of shedding your bacteria naturally, like from your skin. So overall, these concepts are important, not just to understanding what's going on in the environment, but understanding our own health. So this is the One Health concept, which basically says that we need to understand animal disease, human disease, and the environment to better understand ourselves. And finally, before we get into the project itself, antibiotic resistance has really large impacts. So these numbers are from the Center for Disease Control, and it's showing that there's at least 2 million illnesses related to antibiotic resistance per year and 23,000 deaths. And then the healthcare costs can be $20 billion, as well as associated costs for lost productivity up to $35 billion. So a total of $55 billion. So it's a substantial amount of money, as well as a substantial amount of human life and human time, especially in hospitals.
And aren't the people already sick anyway? I mean, it's just they're resisting the medicine.
So it can go both ways. People could just pick up an infection that happens to be resistant, and then they either spend more time in the hospital trying to combat that, or they could die from it. Or sometimes, they already have other illnesses, and then they get an infection. But I think the 23,000 is just from dying from infections. That's like what the primary cause of the death would be.
Are those like septic infections? Deaths?
I don't know the details of what they counted in that number. That would be a good thing to look at.
It becomes a killer.
I'm sorry.
It says it becomes a killer.
Yeah.
You know, alcoholic infection.
Yeah, that's what would be happening. Any questions?
What was the other number?
The other number? Which one?
I got the 23,000.
Whoops.
2 million.
2 million illnesses per year. And they think these are underestimates, so it says at least. Okay, so now that we've talked about all the background, I want to explain a little bit about what I've been doing and the question I've been asking, which is what are the levels of antibiotic resistance at beaches around Cape Cod. So, why are we interested in looking at the beach? First of all, it's because it's a land-sea interface. So we're able to see what's going on from the land as well as what's going on in the ocean because they're meeting up here. And also, like I said before, humans are interacting with it on a pretty regular basis through consumption and recreation. So that represents a potential public health issue. And just driving this point home, we would expect resistance in the environment anyways, even if humans had never ever discovered antibiotics. But they would probably be at lower levels. So it's likely that resistance is enhanced by humans. Okay, now we get to the final field sampling stuff. So these are my 6 sites, and I have 2 in Buzzards Bay, and 4 around WHOI Bay. So first of all, I have Old Silver Beach, which you guys probably have heard of. It's a very popular swim beach. Sorry about that. Then we also have Little Island Beach, which is our isolated, or low impact, because it's relatively isolated, it's hard to get to, and it's in a residential neighborhood, and it's in a private beach. So those are the 2 there. Oh, and then Old Silver Beach would be considered the high impact for these 2 sites. And then we have 4 around WHOI. So the high impact site is at the end of Bayside Drive Narrows in Seacoast Shores. And then we also have 2 what we consider medium impact sites right here at the beach right behind us. And the difference between these 2 is that one site is further down on the beach, and it has a nitrogen removing barrier underneath the beach. This was already in place from somebody else's previous experiment, but we thought it would be interesting to see if we saw a difference between the part of the beach that didn't have it and the part of the beach that did. Right now we're not seeing a difference, but who knows, we might. And that would provide a potential way of maybe reducing antibiotic resistance, potentially. But it was mainly, we looked at it because it was already there. And then our last site is the low impact site. This is by South Cape Beach off of Will's Work Road, which is a relatively isolated site, sort of hard to get there. You'd have to have a higher up vehicle, otherwise you would really mess up your car. So that's why we picked these sites. So what we do, this is the methods flow chart. We go out sampling, then I come back and bring those samples back into the lab and culture bacteria, and then we do resistance testing. So I'll explain each one of these. So first of all, when we go field sampling, these are some of the pictures from the sites around WHOI. So here I'm using an environmental probe to go out and take environmental data. You probably can't see it, but my hand's out like that, so I'm holding something down into the water. And that probe takes environmental data like dissolved oxygen, temperature, and salinity, so we know what's going on and what the physical parameters of the environment. The next photo is me taking a water sample up here. And we take water samples, we take dry sand samples, wet sand samples, as well as shellfish. And then finally, we write everything down. So I'm writing on this data sheet here about the information that we collected from the environment, as well as how many shellfish we found, what kinds, all that sort of stuff. And then I also write down what's happening at the beach. So how many people were there, what they were doing, if people were swimming, if we saw any animals, all that sort of stuff, just so we have that information. So we've been doing this for the past year, and we'll actually have the final sampling next month in May. But over the past year, sampling has thrown some challenges our way, including ice. So there's ice at the beaches, so Becky and I went out and went through the ice and got our samples, which was an exciting experience. It was a lot of fun. So ice doesn't stop our sampling, and we keep trucking on to get them. So once we have the samples, we head back to the lab to culture the bacteria. So these are the 3 different types of bacteria we culture. So the one on the left is Enterococcus, and this is considered a fecal indicator bacteria, so it indicates fecal contamination. And the EPA uses this bacteria to determine beach water quality and safety. These bacteria can be pathogenic, so that's part of the reason why we were interested in them as well. And it's important to know what their resistance levels would be. The next ones are Vibrios, and these are widespread in the marine environment and often have association with marine organisms like squids or other plankton. but they can also be pathogenic. So you might have heard of Vibrio vulnificus or Vibrio peritonelicus, and those can cause gastrointestinal illnesses from eating the contaminated shellfish. Also, the causative agent of cholera, so Vibrio cholera, is in this group too. But there's a lot of ones that aren't pathogenic here as well. But those ones also aren't as well known. And then finally, the last one we look at is the general marine bacteria, which is on the right. So this is a large grouping, basically. So the two on the left, well, the left and the middle one, we use selected media so that we know that we're getting these types. But the general marine bacteria is just like any bacteria that would grow on ocean-like food, essentially. So it's pretty wide open there. But we think this one is particularly interesting because this is what would normally be in the environment and would potentially be the main environmental reservoir of resistance, what's sort of sticking around all the time that can potentially pass resistance to pathogenic Enterococcus or potentially pathogenic Vibrios.
So you culture these 3 different types by putting them in 3 different media to grow in.
Exactly. Yep. 3 different types of food. And then you let them sit in different temperatures, and then they turn out like this, basically. So then what we do is I pick isolates off of these. So to start, for example, the Enterococcus, can you guys see the little blue dots? So those are the colonies. So I'll pick up to, depending on how many are there, 9 colonies per plate on those ones. And then for the Vibrios and the marine bacteria, you can see that there's different colors. So here you can kind of see this yellow and the green. This one, I'm not sure how well you can see it, but there's yellow, white, there's different colors of pink, all that sort of stuff. So what I end up doing is I pick 3 of each different morphology. And this process sounds pretty simple, and it is pretty simple. However, it's incredibly time-consuming. And I'm only pointing that out so later on, when I show you not as much data as I would like to have, that you're sympathetic to my cause. So overall, I've collected a lot of these. So there's 500 about for each of the Enterococcus and the Vibrio, and 1,500 for the heterotrophs. And these are all stored in a freezer, and then when I'm ready to test them all, I'll just pull them back out and start chugging away. So that's what we'll talk about next, is the resistance testing. So this is a little schematic of what we do with the resistance testing. So imagine the circle is one of those petri dishes that I just showed you, and there's media on it that the bacteria like. So what you do is you take the petri dish and you swab over it with the bacteria to create a lawn. So that's what the dark beige back is supposed to be. That's all bacteria that's really dense and really close together. And then what I do is I set down these little white discs, which are here. And the little white discs have antibiotics in them, and they're embedded in the antibiotic with a known concentration. And you can just buy those commercially. So we put those out, and I just have the names of the antibiotics written around this as an example. And then you put it in an incubator at a given temperature, let it sit overnight. And then what I do is when I come back in, you take a ruler and measure the zone around it. So this is the other, I guess, white color, gray, around here is the inhibition zone. And that means it's showing whether or not the bacteria was able to handle the antibiotic. So if there's a large zone, that means it's sensitive, right? It wasn't able to grow, so that's showing no growth. But if it grew all the way up to the cell, or to the disc, like this one, that means it's resistant. Does that make sense? Okay. And you measure the number because the number can be different for different antibiotics. And there's standards for all of that, so basically I take the measurement and then I look at the standards, and that allows you to determine if it's resistant or sensitive. So this is actually a plate that we did, and I just wanted to show you so you know what it looks like. So the beige color is the bacteria. Here we're seeing a resistant disc, and then this is one with a large zone that's probably sensitive. But here you can also see that there's not a lot of growth around that one, but that probably also is resistant, but you have to check the numbers. So I just want to quickly point out why we picked these antibiotics. So these are the 6 antibiotics that we studied, and there's just some of the different uses that they can be used for different infections. So some things that might be interesting are their diverse start dates. So if you look at oxytetracycline, it started in the 1950s, and it can be used for anything from throat irritations to syphilis. But clindamycin, on the other hand, is one of the older ones, and it began use in 1989, but can also be used for diverse things like malaria or acne. One of the ones that might be particularly interesting on the Cape is doxycycline, which is often used for Lyme disease. And even though these started at different times, just because they're older doesn't mean that we don't use them. Doxycycline and oxytetracycline are actually used very often, still today. So, remember when I said the picking process takes a long time? This is where that comes into play. So, I really only have data at this point for 316 of the Vibrios, and I'm working on the Enterococcus and the heterotrophs. So we're just going to talk about the Vibrios for the rest of today. However, within that grouping, we found that 94.3% of those 316 were resistant to at least one antibiotic. So that's a pretty high amount and was higher than what I had anticipated that we would find. So now I want to explain what we learned so far. And I also want to keep in mind that the data is relatively new and we're still getting more data. So I haven't done any statistical tests on this data, so I don't know if anything's significant or not. So it's just trends, things that might be happening. No for sure's. So to start out, I just want to show the resistance and what we saw. So on this axis, we're showing resistance to a number of antibiotics. This means that, like, for example, a bacteria isolate was resistant to 2 antibiotics. It doesn't have to be the same 2, it's just any 2 of those 6. And then up here on the vertical axis, we're showing the number of bacterial isolates. And as you can see, there's not that many that were resistant to 0 antibiotics, probably about 30, let's say. And then there were a lot, over 100, that were resistant to 1 and 2 antibiotics, and then it declines again to about 70 for 3, and even less for 4. For 5, I think this is only 1 sample that was resistant to 5, and luckily enough, we didn't have any that were resistant to all 6. So here we're seeing a very small potential seasonal pattern because overall the resistance levels are pretty high. The data that I looked at was from June to December, and we've been collecting the other data, but I just haven't done it yet. So soon enough, we'll have all of that together for a full year. But here we're seeing that June and July have very high levels, and 100% of the isolates were resistant to at least 1 antibiotic. Oh, I should point that out as well. For the rest of these graphs, percent resistance is defined as resistance to at least 1 antibiotic. So we have 100%, then it drops down to about 86% in August, and then picks back up and drops down again a little bit in December, but not as much as in August. Again, these are relatively small changes, going from like 100% to 86% is a substantial amount, but it's not huge. It's not like we went to 10%. So it'll be interesting to see what the rest of the data looks like to really figure out if there is a trend or not.
It's only 1 year.
That's true too.
You know about next year.
Ideally, we would be sampling for a long period of time, right?
It would have more meaning if you had.
Exactly. Sadly enough, not possible. In terms of funding, time, all of those sorts of things.
Actually, her study is the only study that's looked at it for more than 2 months at a time.
Oh, okay.
That's good.
Yeah.
Well, how do you have any way of accounting for the drop in the resistance in August, September? Do you think that's an anomaly?
I'm not sure yet. So it's hard to compare it because there haven't been other studies that have looked at seasonal patterns. And in general, people just go out to a site, they measure it one time, and they write it up. So So I'm not really sure. That said, I found a paper that looked at antibiotic resistance in clinical patients and clinical infections in hospitals, and they showed a drop down in the summer too. Theirs was way more substantial though. It was much higher drop down. So I thought that was interesting, and they sort of attribute that to the fact different infection levels over time. So, but yeah, again, it's 1 year, and we don't have a full data set yet. But it's something to think about, and I like thinking about that stuff. So I pulled these out by the Buzzards Bay sites and the LaQuoit Bay sites. And if you'll remember, the LI site is our low impact, and then Old Silver Beach is the high impact. So those are the two that showed the biggest, most substantial difference, but still it's not that much. We're probably going from 94% to about 86%, which is relatively small. But then interestingly enough, most of the sites around LaQuoit were pretty similar. And actually the most impacted, what we consider the high impact site, which is right here, is the lowest. So that's sort of different than what we're seeing here. Again, sort of want to get all the data to really look at it. But one of the things we're potentially thinking about is the water in these environments. So in Buzzards Bay, it's a little bit more high impact. There's probably more flushing going on. Whereas in LaQuoit Bay, you're having the one inlet, right? So water might be sticking around longer. Maybe similar water is going to all of these sites compared to these two. Not sure about that, and it's something that we'll have to look into more. So this, I think, is the most interesting graph. So we're seeing that the antibiotic results really vary substantially. So here's the antibiotics there, and again, the percent resistance. And I also added in the years that the antibiotics were introduced because I think this shows somewhat of an interesting trend. So it seems like antibiotics that were introduced earlier for human use actually have less resistance compared to the ones that were introduced later on. This is counterintuitive to at least what I would have thought. I would have thought there would be more resistance if we've been using it for longer. So yeah, that's interesting. I'm not sure what to think about that, and I'm really excited to see the rest of the data as well as compare what I found within the Vibrios to those general marine bacteria and to the Enterococcus to see if they show similar patterns or maybe it's different and totally bacterial type dependent. I'm not sure.
Does that mean the sharp ones, if you've got something you want to kill, then you should take one of those with the sharp?
So it says that for marine bacteria, well, I don't want to make any recommendations on what types of antibiotics you can take. I think you should take whatever your doctor tells you to take because it depends on what infection you have, and different antibiotics work for different types of infections already. So yeah, I can't tell you any recommendations about that. So lastly, for graphs, I wanted to point out the sample type differences. So this is wet sand, dry sand, and water. And as you can see, they're pretty similar. But again, this is something that is counter to what I would have thought. Water is showing the highest amount, though granted by probably 2 to 3%. I would have thought that the sand would have, because in general, sediment has a lot of antibiotic production going on. So I would have expected those to be higher than the water. But again, it could just be different than what I was expecting. This could be just for that time period. Who knows? We have a lot of work left to look into these things. So quickly, I wanted to compare the resistance we found in this study to resistance in other studies. So overall, I'm finding more resistance in Vibrios than other studies have, but there are some caveats to that. So one is that other studies are generally done in the developing world, and we would expect that their environments would potentially have different types of Vibrio species than we have here. The studies that I've looked at also have smaller numbers, so they do maybe like 100 samples, and I've done 300 already. So that could be another difference. And then one of the biggest and hardest thing to tease apart is that they're testing different antibiotics. And you would expect that given a different antibiotic, you would get a different resistance amount. But the way the data is reported is it's hard to pull apart the different resistances. So that's something that remains to be worked on and remains to be compared. So what are we doing next? So I'm working on resistance testing for the rest of the Vibrios, as well as the Enterococcus and the general marine bacteria. And then what we want to do is identify all of the bacteria by genetic analysis to figure out exactly what species, essentially, those bacteria are. Because depending on what the diversity is out there, that could be playing into how much resistance we're seeing. And then finally, I'm looking into quantifying the levels of human impacts at these sites. For right now, we put them as like high impact, low impact, medium impact, but it would be really nice if we had quantitative numbers to pull in with that to better figure all this out. Does anyone have any questions so far?
I was wondering, you were mentioning earlier about bacteria transferring antibiotic resistance to other bacteria. I was wondering, since there's a lot of dissolved organic carbon in the ocean and some of that's presumably pharmaceuticals, whether that is a pathway for introducing antibiotic resistance in marine bacteria.
I hadn't thought about that. That seems reasonable. Yeah, I mean, one of the other pathways is like kind of the antibiotics residues or actual antibiotics that get into the environment as well. So that seems like it could be a reasonable pathway too. So to sort of end, the last part is where does this research fit into the larger picture? So far, these are our general study goals in looking at the marine environment. So the first two we're doing within the study, and the last two are things that we're interested in looking at in the future. So we're coming up with a better understanding of the marine environment and what we expect in terms of resistance out there. We also are looking at if there's links to human impacts, and that's why we want to quantify the human impacts at each of these sites. Hopefully what that'll allow us to do is to determine if there are health risks, and if there are health risks related to this, if it's necessary to make changes in how we use antibiotics.
We know that already. We use too much of it.
I agree with that. Yeah, but I want to have my data back me up with that, you know? I get where you're coming from. And then antibiotic resistance is a global and national problem. So I just wanted to point out that these organizations are working on it and are very interested in this as a global problem. So the World Health Organization, the White House, and the CDC have all released reports on this relatively recently and are trying to take action to combat this. Generally, they look at resistance within hospitals, but it's still necessary to continue doing this work. So now that I've told you that there's all these problems, it seems a little overwhelming, it seems like it's out of our scope, there's a couple things that you can do that I just want to point out, and then it doesn't sound so doom and gloom either. So one thing is to preserve the integrity of antibiotics. So because of structural differences, if you have a virus, so like the flu or the cold, if you take an antibiotic, it's not going to help you. And as we talked about earlier, it's actually could potentially hurt you because you're killing off your good bacteria in your gut. So you shouldn't ask your doctor for antibiotics if you don't have an infection. That said, if you do have an infection, you should take your antibiotics and you should take all of them because that helps to make sure that resistance doesn't occur. And another one is if you're able to buy antibiotic-free food. We can make decisions with our wallets, and a lot of places actually have started to make changes because people have demanded them. That said, I know this can be expensive and not everyone's able to do it. It's just something to think about. And then finally, to wrap up, in conclusion, we're just beginning to understand resistance at our beaches. It'll be a while before I've collected all the rest of my data, tested it and analyzed it for our complete data set. But despite that, it's important to remember that we as humans impact our environments. And antibiotic resistance is a major problem that needs to be understood to better protect human health. So overall, resistance is something to be concerned about and to continue studying and think about, but it shouldn't stop us from enjoying our beautiful beaches and beautiful and safe beaches that we have around us on the Cape. So that's all I have for you guys this evening. Thank you for coming. And then finally, I just wanted to point out all the people who have helped me, including my advisor, Becky Gass. Many of my friends that I roped into field sampling with me, 2 of which are right here, and my funding sources as well. I can take any other questions that anyone